Back everybody here to the concluding panel discussion for this meeting. Really excited to come to some next steps, and I'll talk about that after all of this is done. But let's maybe start with a brief round of introduction of our panelists here for this final session. And Stephen Carpin, you're up first.
Great. Yeah. Thanks, Joe. Good to be back, everybody. I'm still Stephen Carpin here. I'm the Senior Director of Regulatory Affairs at Breakthrough T1D.
Elizabeth?
Thanks, Joe. Pleasure to be here. Elizabeth Niemöller. I'm a physician with a long-standing history in diabetology and saw many patients at multiple stages of type 1 and type 2 diabetes before I joined industry. Now I'm heading the clinical development department for diabetes, cardiovascular, and metabolism at Sanofi, focusing on these days on the challenges related to the development of disease-modifying therapies for autoimmune type 1.
Great. Jason?
Thanks, Joe. Appreciate it. I'm Jason Gallia. I'm an endocrinologist and clinical trialist out of the Joslin Diabetes Center in Boston.
Great. Dr. Siegel, please.
Hi, my name's Jeffrey Siegel. I'm an internist. I'm office director in the Office of Drug Evaluation Sciences, in the Office of New Drugs and the Center for Drugs at FDA. Our group is responsible for qualification of biomarkers and clinical outcome assessments, among other things, across all of OND.
Great.
Looking forward to the
much.
discussion.
Yeah, Matthias, and you're on mute, so you'll have to come off mute here. Thanks.
Matthias von Herrath, very happy to be here from the Global Chief Medical Office at Novo Nordisk. I've been with Novo since about 13 years, and also from the Diabetes Research Institute in Miami. It's my personal passion to find better solutions for patients with type 1 diabetes, and I'm excited about the workshop and this panel. Thank you for having me.
Thanks, Matthias. Yeah, Dr. Sheretz.
Hi there. My name is John Sheretz. I'm the Director of the Division of Diabetes, Lipid Disorders, and Obesity in the Office of New Drugs, Center for Drug Evaluation and Research in FDA. And I'm an endocrinologist by training.
All right, well, I think we've got the right group of folks here. So, John, I'm actually going to start with you. You've been listening to the discussions over the last few days. Would really be curious for you to kick us off and sort of provide your perspective as to where this question of C-peptide as an endpoint for clinical trials in new-onset T1D stands. What are the strengths? What are the weaknesses? And ideally, what more can, or rather, how can the T1D community help regulators with the decisions they need to make?
Sure. And I think, I guess your question is, where do we stand with the use of C-peptide as an endpoint?
Yeah.
So, what I would start by saying that I think that the observational data links C-peptide to glycemic control, reduction in hypoglycemia, and long-term clinical outcomes as part of natural history is very strong. That is, patients with more severe disease who have more rapid decline in C-peptide have worse glycemic control and worse microvascular or macrovascular outcomes, whereas patients with less severe disease have a more benign course. And even when we account for confounders such as age, such as duration of diabetes, C-peptide is a good predictor of the natural history of the disease. And I think the rash in clinical trials, we have seen with drugs that preserve C-peptide. We have seen evidence that there is a pharmacologic effect with them. And consistently across trials in drugs where C-peptide is preserved compared to placebo, we see reductions in insulin usage, which is something we would expect. I think the big question in the clinical trial data is what that apparent treatment effect, how it translates to clinical outcomes. And I think there's big questions about why when we see these findings, why we aren't seeing effects on parameters such as A1C, such as hypoglycemia rate, or other things that are measured in the trials. And I think there's a lot of uncertainty around that. I mean, I think it's possible, and in some cases, that it's simply an issue of trial power, and that's been floated as an idea. But I think other considerations could be that the magnitude of the effect on the drugs may simply not translate to large clinical effects in current context. And I think the third possibility, which we hope isn't the case, is that perhaps the intervention is simply occurring late in the course of the disease, and even large magnitude effects aren't sufficient to impact here. But I think to try to tease these things out, we saw some of the interventional analyses, both in the Taylor paper that was published a couple of years ago, and in some of the data presented today. And well, I think this is the place where we're interested in tying C-peptide to A1C. I think there are some really important limitations in the data that we've seen, and I'd be interested in knowing more about the data in order to resolve some of the questions. So in the Taylor article, I think one of the big concerns was that the way the analysis was done is certain trials were included in the analysis as well, actually, let me go into the details. I think that one of the issues is that there continue to be some limitations with some of the data that were presented that I have questions about, and I can come back to later. But I think that that barrier, the lack of ability to show benefits on clinical outcomes in trials, and some of the limitations of the analyses prevent us from being able to use C-peptide as a surrogate endpoint for approval. But I think that the evidence that we do have does suggest that using it as as what we consider a reasonably likely surrogate endpoint that needs to be confirmed by other data is where we stand right now.
Okay, I will want to go to a number of the other panelists here just to kind of respond and add their own thoughts. But I see, Stephen, you've got your hand up. So why don't we start with you?
Yeah, thanks. And John, thanks for the comments there. I think, you guys over at FDA have been pretty clear, at least since the 2021 workshop, that at the moment, we've got quite a lot of data suggesting that a lot of those are phase 2 studies that, as you say, weren't really powered to look at those clinical outcomes. They were powered to see C-peptide. So let's bring all that data together, take a look at what that tells us when we have a couple thousand subjects rather than maybe a couple dozen or at best a couple of hundred, and see what we can really learn about the causal relationship through that interventional data. I think, as you've kind of suggested, that first Taylor analysis, perhaps there are some concerns with what that tells us. There's more that we need to kind of dig into that TOMI database to understand exactly what's going on. But I think, the points you're hitting on here, I think where we are today is a result of more or less what we heard in 2021, that we had this gap in understanding what the causal relationship really looked like. There's no doubt that C-peptide is prognostic. We see very clearly in the observational studies that relationship. The question is it predictive? And so being here, being able to really engage on what this TOMI data tells us, what the gaps in that are, how we fill those gaps, I think that's really the foundation for the conversation that we're wanting to have.
Yeah, so Elizabeth, I'll maybe turn to you next to add your perspective here.
Thanks, Joe. I think during the last 2 days, we've heard a lot about C-peptide. And I think we all acknowledge that C-peptide matters. It's very important. And we all acknowledge also that we face this high unmet medical need. It exists, and it exists that we have, after a century of insulin treatment, now, I would say, almost the obligation to move forward to bring disease-modifying therapies to the patient. Thinking back, insulin treatment basically tackles the glycemic situation, which is a result of the autoimmune process, which destroys the beta cell. And the disease-modifying therapies that we want to move forward are supposed to break the underlying immune process. C-peptide is a measure of the beta cell that is destroyed. While when we are looking at glycemic parameters, we are looking at the effects of the underlying background treatment, insulin. And I think we need to acknowledge this, that we have eventually here a new treatment paradigm, I would say. And for that, we also need to acknowledge that we need to look at new endpoints. I do believe we all agree that C-peptide is very important. The evidence is going forward. The Tommy paper is underlying. We saw also the great analysis from Helen. And I think we are now at a stage also in execution or in the next step following the 2021 workshop that we need to move forward and consider C-peptide eventually as a future surrogate, because we have seen so many new data now. And I think we need to find a way how we as physicians, as researchers, together with regulators, can identify the path forward in order to bring these new treatments to the patients. Thank you.
Yeah, Jason, I want to go to you next. You've been involved in a lot of studies over the years, so I think your perspective would be valuable here as well.
Yeah, and just to follow up what Elizabeth said, to me, C-peptide is just clean. It's a measure of the beta cell without any measurement of underlying what's going on, the insulin treatment, the activity level, the diet. So I can have a patient who can get any A1C you want, but they can't get you any C-peptide that you want. The C-peptide is just intrinsic. And so because of that, I think the C-peptide really offers value that we're not seeing in a lot of these other measurements. And so just the purity of that is so important to me as a clinical trialist, because it measures what I want to measure without this noise around it.
Yeah, Matthias, I'm going to turn to you to add, and then I may come back. I have another question for Elizabeth, but yeah, watch your mute. You're on mute, Matthias.
My biggest quest here is to see how we get there in the least painful way to open this recent onset diabetes treatment space, because I think we can probably all agree that more safe, ideally very safe drug activity in that space will give patients ultimately more options. It will push things also to earlier stages where the trials are maybe slightly more complicated. Increase our learning, including what can be combined, what should be combined, and what could we end up with combination therapies, but we need activity in this space. And I guess there are several things we could do. One is we could come to an agreement that certain C-peptide levels per se are enough, maybe. The other thing that we might be able to do is to say, rather than getting into an argument of how a treat to target trial where HbA1c is influenced, as just said very eloquently, by many parameters, can we look out further? Can we get a conditional approval and then bring back data that the community needs, but they are further out? Of course, that has its own problems, but maybe that's a compromise we can consider in the present situation. I just want to put these things out here. Of course, not particularly easy. So that's where I'm with my thinking.
Yeah, John,
really impressed by the wealth.
Yeah, sorry, Matt, you're cutting in and out a little bit there, but I think John maybe give you a chance to comment here, and then we can come to Stephen, and then I still have a question for you, Elizabeth. So yeah.
Sure. I just wanted to get back, respond to Jason's comment about how C-peptide is a clean biomarker that's very closely tied to beta cell function. I think that's true, but in my regulatory world, I deal very much with biomarkers, and this is the whole issue with any biomarker. And I'll give you a good example of something where I work. Lipoprotein little a is strongly associated with cardiovascular disease. It's an independent predictor of cardiovascular disease to LDL. However, in the past, drugs that reduce Lp(a) haven't consistently shown benefits. Now, in current practice, there are some new nucleotides that inhibit production of Lp(a) and lower levels by up to 90%, and they're under trials to evaluate if they have an effect. And if they are approved, that's revolutionary. But the issue is, even though we know the association is strong, the magnitude of the effect really matters. And I think that is one of the challenges in interpreting the changes that we're seeing in these drug trials, which are quite different from what we're seeing in other areas. For example, I think the example of islet transplant is brought up. I mean, the magnitude of difference from bringing people with essentially no detectable C-peptide to levels that are higher than what we're seeing in this level, that's different than what we're seeing in these drug trials. And I think it's important to define whether or not these small changes do translate to meaningful benefit. And one thing I'd add is that I think that the context of use really matters. The other area that I work in is in the LDL levels. And contemporary trials where we studied agents to lower LDL have to consider what the background therapy is. We don't conduct trials in people who aren't on statins. It's first of all, because it's standard of care, but second of all, because it's unethical to enroll people without therapy. So drugs that in the past had potentially showed promise in reducing cardiovascular disease, like niacin, when they're studying on a background of LDL, they don't show additional benefit. And I think that even though there is a huge unmet need, despite the optimal therapies that we have right now, you need to evaluate the therapy in the context of available therapy. So if you have automated insulin delivery or insulin pump and CGM, and that has a big impact on glycemic control, and that to some extent obscures the effect of the disease-modifying agent, we have to understand what the effect is in the context of current care.
Yeah, Stephen, give you a chance here to add your thoughts, and then John, I want to follow up on a couple of those last comments.
Yeah, John, and to start there, I think that's part of why it's so important what we've heard the last couple of days about thinking about other ways to show clinical meaningfulness, looking beyond just A1C, beyond just hypo, to think about what it really looks like day to day for patients with type 1 and how we can make that day-to-day experience better. Because we know even with the best AID systems that are out there, it's hard. It's not easy in many different ways. And what we have now just isn't frankly enough there. The other point I wanted to touch on is we've kind of been dancing a little bit around the idea of what's the threshold, right? How much C-peptide do we need for it to be enough change, enough preservation in C-peptide? And I guess I would pose the question, do we really need a threshold? Do we need to have a defined level that says this is the amount that's enough? Anything above this, we're good. Anything below this, we're not quite there. Or is it enough to say we understand the relationship? We can clearly say, based on the data that we have, this much C-peptide is expected to have this much benefit. If you have more than that, this is how much more benefit we'd expect. And be able to just say then, in the context of a specific individual drug, we can look at how much benefit comes from however much C-peptide preservation we see in that specific trial, put that then next to the risks of that specific agent and say, without any specific threshold, we can look at our relationship between clinical outcome and C-peptide, measure that very directly and clearly against risks, and have our answer about whether that drug is approvable without having that specific cut point that says, this is enough, this is not enough.
Now, so John, want you to respond.
Oh my, yeah. Well, but I think that's the information that isn't there. I mean, it's when you say that this amount of C-peptide preservation is expected to have this effect. Well, I don't think we know that from the interventional side. You're inferring from observational data, but observational data is different. A patient who has a slower immune response, and I think of a middle-aged adult who develops type 1 diabetes, yeah, their glycemia is better, their long-term outcomes are better, but they're very, very different from a 5-year-old who progresses rapidly and has a low C-peptide within a few years. So I don't think you can make that inference that a drug that makes these incremental differences translates to that difference between those two patients. And even within the 5-year-olds, there's heterogeneity. I mean, disease severity varies. I think that the issue is that we don't really know what a certain amount of preservation of C-peptide is doing in that direction. We understand it observationally, but that is it's a different context than what the drug does, what a drug can do.
Yeah, I think that's what we tried to show in that Taylor analysis of the Tommy data. And from what I'm hearing from you is there's some things we need to address in that data set in the way those analyses were done to really be able to answer that question, again, with the interventional data itself. Joe, I think there's some plans in the works to help get that data from Tommy to the FDA. So you guys are able to get your hands on it.
Yeah, that's already been set up actually. So,
which is great. And so hopefully that's our answer, John. Hopefully when we can look at that data, look at it in, the way that you want that data to be kind of taken apart and looked at, we are left with a really clear understanding of what the interventional data tells us. And therefore we have an answer there.
Right. And just, so you understand like where I'm coming from. I think it's a little hard to interpret the data in Taylor because the methods aren't entirely transparent, but the types of things that I have concerns about. So one is they looked at 31 trials, and of those 31 trials, 10 were excluded and 21 were left. And of the 21 that were left, 9 were included as positive studies, and the remainder were negative. I may be wrong. It's either 8 or 9, which meant that the analyses took place on about 800 patients, and 1,900 patients were excluded from analyses. But when I looked granularly at certain choices in the analyses, it raises concern. So for example, there were a few trials where there were multiple arms. And the trial as a whole was considered a positive trial. And I'm not sure, but from looking at the numbers in the analyses, it appears that subjects from the arms that would have been a negative trial if they were a standalone trial are included in the analyses. And I don't know which direction and what that may introduce to the analysis, but I know that is a limitation there. And like I said, I think, but actually I think the way the analysis was done in Taylor probably isn't the way to go. I think the analysis we saw here, the meta-regression, where you look at all trials and you look at all levels, that's really the way to go. I mean, again, we don't have the background on what was included there, but what I do note is that there were 17 data points on that. So I know there were 31 trials that study C-peptide and A1C. Maybe it's reasonable to exclude some of them, but of the 21 remaining, I also know that there are ones that study more than one agent and have more than one arm. So it seems like we should be able to have more data points to do those analyses, and maybe we can understand those data a little bit better. So I do have some interest in that area, but like I said, we'd be interested in seeing that.
So, John, I want to go back, to a point you made earlier, right? That anything we do has to be on the background of standard of care, and obviously, that's not even an option in type 1 diabetes. Insulin's life-sustaining, so that's a given. However, I think we have seen from some of the data, for example, from T1D Exchange and other places, that and I mentioned this in the earlier session, this idea of the Hawthorne effect, that you can see up to 0.5% better HbA1c in clinical trials just by the fact that people are in clinical trials and getting probably the best possible care that can be obtained. But then when they go back, when they leave the study, they go back to their regular lives, they're kind of on their own with their own endocrinologist to manage the disease. And again, the T1D exchange data highlights the fact that at least here in the United States, we're pretty far away from where we'd like to be. And so this kind of raises this question of, are we hamstringing ourselves in the way that we're doing the trials today? And is there a better way to do that, to tease out potentially what the benefit is in that real world setting? I mean, that is a confounder. I think you'd agree, but are there ways, maybe from your guys' perspective, are you open to a more what I referred to earlier as more of a real world type of design, incorporate some of those elements into these studies so that we can at least remove that effect from these studies, or reduce it?
Well, I'd say two things, but I think the important thing to recognize is that there are two different concepts. So one is, does optimized care in the setting of a clinical trial alter the direction of the effect? And I say, well, in that case, no, because that's the whole point of randomization. Subjects are randomized to placebo or the drug. Both of them are getting optimized care, and both of them will have improvements in A1C as a result of that care. The second issue is a different one, and that is, does that affect the magnitude of effect that you can detect? And absolutely, it does. And I think that is a big challenge with studying A1C in trials. And I think that's one of the reasons we've been thinking about, what are some other metrics that we can look at? And again, when we talk about disease burden, well, if it's a patient who's on multiple daily injections, is it, freedom from prandial insulin or reduction in the prandial insulin doses? In patients on pump, I think it's more challenging to get a direct effect, but what can you measure there? And I think this idea of trying to develop an instrument to study what disease burden is. I think that was something that would be interested in. And maybe, this is a place to tee up Jeff, since that's his sort of his area, but what are the things that we need to do to develop an instrument like that? And I think one of the important things is, like, you can't just take an off the shelf general health quality of life measure. You need something that's specific to type 1 diabetes. You need to measure concepts that are meaningful to the people that are the intended population. And the instrument has to be sensitive enough to change to be able to show a difference between treatment and placebo. But maybe I can hand it over to Jeff to see if he has thoughts on that.
Yeah, Jeff, I'd appreciate your comment. And then Stephen, I'm going to come back to you because I know you guys are doing some work in that area, but Jeff, maybe you first.
So just want to make sure I understand what you want me to comment on. You want me to comment on developing patient-reported outcome measures that measure the burden of disease that could potentially be impacted? Okay.
Yes.
So the framework then this should be tried out, especially in longitudinal trials to understand whether it's sensitive to change, test pretest reliability, and so on. So this would be a way to identify in groups of patients how the disease burden impacts them and particular ways that the drug treatment could impact them. I have to say, though, that as a scientist, as an immunologist, I think that we shouldn't focus exclusively on these aspects. I think there's more to be done in controlling the severity of the disease and the pathophysiology. For example, in the curves that I've seen from the TAMI data, the positive trials seem to show an improvement in C-peptide initially, and then a subsequent decline. And when I look at the slope of decline, it seems to decline in parallel with untreated patients. So what it suggests to me is that there's ongoing damage to the beta cells that's worsening the disease. If you can get a truly disease-modifying treatment that can not only transiently improve C-peptide, but change the slope, I think you might have a treatment that could be very beneficial. And the benefits to the patient, I would hope, would become obvious once you're really preserving beta cell function. Hope that's helpful in terms of a number of different ways that you can think about endpoints for this important condition.
Yeah, Stephen, let me go to you, and then I'm actually going to go to Matt on sort of this immune points that Dr. Siegel was raising.
Yeah, and Jeff, thanks. I couldn't have hoped for you to lay that out so clearly for the PRO measure, especially because that's precisely what we're doing now with the ongoing PRO work that we've kicked off at Breakthrough. First is we have together a draft concept model of type 1 diabetes based on what's in the published literature, and we'll soon be starting the concept elicitation studies to really do the qualitative work that needs to happen to solidify what are those important concepts to patients. From there, our plan is to map out those important concepts to, in this case, what's going on in the new-onset space, what's most important to patients, what concepts do we think the therapies we have are able to impact, and then look at, for the existing measures, most of which probably aren't up to kind of regulatory grade right now, can we do a little bit of work to bring those up to regulatory grade where they can inform our decision makings in drug development? And if not, do we need to think about developing a new measure from scratch that is going to be of enough quality to answer those questions? I think before we leave the topic, the question I have about that for you, Jeff, and John, for you as well, is it takes a lot of time to develop a new PRO measure, to kind of go from beginning to end to do this work. We're talking at a minimum, probably 3 to 5 years, best case scenario, to get to the point where we have a measure that is high quality. And that's not even if you're considering implementing that into enough trials to be able to get that longitudinal data you need to see if that measure holds up in the context of a clinical trial. The message we hear from our patients is we can't wait. We can't just sit back and kind of wait for that to happen in order to make progress. I don't think that anyone here is saying that that's the only way we make progress and that that's what we need to do. But I guess the question I have is, how do we think about doing that as quickly as possible? How do we make sure we're meeting the evidentiary standards that we need to for developing a PRO measure that can inform regulatory decisions, but do that in a way where development doesn't have to stall while we're working through that?
Yeah, Matthias, let me go to you, and then we'll come back to John. But so one of the things that Jeff mentioned was this idea of the immune intervention, and I think one of the things that you've talked about in the past, which I have mixed feelings about this, but I'm unfortunately very familiar with rheumatoid arthritis. The anti-TNFs were game changers in this space, but there's a lot of people that do not respond. And so, yes, Jeff, what you're saying about the trials is true if you look at the aggregate data, but there are people within those studies that don't lose any C-peptide over the time that they're being treated in a number of these clinical trials. And so, Matt, I know you've seen this. You know that there's always subgroups that do really well on these interventions. You don't generally see those in the placebo arms. And the other thing, as I mentioned, in the previous session too, we're already starting behind the 8 ball, so to speak. We're not intervening in these folks sometimes 3-4 months after their diagnosis. That may be less important in adults that have a slower rate of decline. Jason, I know that's a point you've made, but in the ped population in particular, their C-peptide is dropping, very, very quickly. So, Matt, I wonder if you'd kind of just give your perspective from the immunology side of the equation, and then we'll come back to John and then Elizabeth.
Boy, there's a lot to unpack here. So we can probably, Elizabeth can probably speak well to the responder issue because they have very good data on binding CD3. And I'm gonna, that there's no doubt in my mind that we have to look at the responders. I wanted to bring up 2 things in terms of the immunology and the continuing destruction you raised in that we all see throughout the trials, Jeff. I think my notion has been, when we have studied the pathology in the human endpoint samples now for 15 years, this is a disease of the beta cell and the immune system. There's absolutely no doubt. That's why I continue to believe in combination trials to get the best benefit. And I think there is a reinitiating component from the beta cell that re-triggers immune recognition. And we have to ideally address this. But hence my previous comment, the more activity we get in this space, the easier time we will have to combine drugs, might at least have a conditional approval. I wanted to also, that's my last point, challenge one concept, which I worry about a lot, which is, of course, it's a valid concept to say the natural history of the disease is not the same than when you have interventions. I mean, that's a valid argument. This is not arguing about this. But I'm worried that we end up making perfection in some ways, although well thought out, the enemy. And that's the issue, and I'm very interested in this and what other things of tipping points. We are not good in predicting individual tipping points, not in immune markers, not for different endpoints where we could say hypos and HbA1c and retinopathy, they may have different tipping points. We can't predict tipping points. We can't do it in climate change, and we can't do it well in human individuals. And it was brought up before that we would like to ideally say, well, Matt, you have this type of entry criteria, your trajectory is Y, your tipping points are Z, and this is what we predict for you. That's making things really perfect, frighteningly to me, because I'm not sure we can get there easily. We could try. My suggestion remains to collect some of these things post conditional approval, as long as the burden is not too high, and look in the longer future, and maybe take some things out where we know that the tipping points or the threshold levels are already solid enough in terms of organ preservation, we can be pretty certain. So these are my 3 ideas, but I'm passing the responder analysis and comments to Elizabeth or somebody else who would like to comment on that.
Yeah, maybe Elizabeth will let you talk about the responder point, and then we can come back to John here.
Well, I don't want to hijack John's comment. So basically, yes, of course, there are always people who respond somewhat better to these treatments. And we know that from other areas as compared to others who do not as good respond, right? But what we need to make sure in the overall entity in the setting of a clinical trial, we have seen an overarching beneficial effect, and in many studies. And I think we are able to measure this effect already now. I agree, we need to develop more measures like the PROs, as mentioned by Stephen. And I think it's a very important thing to do. I would think that also we need to think, if we want to move this forward with regard to C-peptide, how we standardize our measures. We heard yesterday the session about the different assays, and I do believe this is also something that we need to consider in the context eventually of the TOMI analysis. But it made me think a lot, and I think we need to move forward in this direction. I would think even jointly, in order to develop these standards, to make it reasonable and acceptable. Because the fact that C-peptide matters, I have the impression we are not debating on that. It's a question on how much does it matter, right? And I think this is where we need to move forward and come to the next level of the discussion. Let me stop here.
Yeah, John. I mean,
Okay,
you've got a lot to cover.
Yeah, I
so.
know I had a lot, and please remind me if I'm missing something. I think the first thing I wanted to touch on was the responder analyses. I understand why there can be interest with that, but I think that is a very challenging area. I mean, I work in rare disease, but I also work in highly prevalent disease, and it if you look at a waterfall plot of a treatment arm from a trial, there are people who, and I'm talking about like something very objective like LDL. There are people at one end who have a 100% increase in LDL, and people at the other end have a 50% decrease in LDL. And if you look at the placebo arm versus the treatment arm, the pattern is the same. There's just a shift in where the inflection point is. The placebo is centered at 0. The active treatment arm is centered at one end, and more patients are responding. But I think you can't look at the person with the most extreme response and say that they responded to drug better. You can't distinguish natural history and intercurrent events from that specific person's treatment effect. So I think that while responders are appealing, I think it may not give you the information that you think you're getting. The thing I wanted to come back with Stephen is that, what he raised the point of how difficult it is to develop a PRO and how long-term it is. And he's right about that. But I think one of the other things that occurred to me as I thought about it is that I think a big challenge in some of these drug trials is that you also have to get a sense of how you can account for biased observation in a PRO, because if you're getting an immune therapeutic where subjects know they got the drug because of the side effects, that can bias the results on a PRO. So maybe that needs to be consideration of development, and that's why I think some of the more face valid treatment burden endpoints, like something like reduced doses of prandial insulin, reduced correction doses, those might be a better place to look at in terms of trying to evaluate disease burden. And was there another topic that had...
I think that covered it, but if
Okay.
If somebody is recognizing something we missed that they want to field up, otherwise I'll turn to Jeff. Yeah, and then I'm going to go to Jason after Jeff. But Jeff, please go ahead.
Thanks, Joe. So I want to back up and make a couple of general comments. I'm going to be repeating things that other people have said initially, but I think you'll see why I'm trying to provide this framework. So obviously, we approve drugs based on a demonstration that they improve the way patients function, feel, or survive. But sometimes it's impossible to measure that directly. And then we turn to surrogate endpoint biomarkers like C-peptide is the one that's being proposed here. And here we're measuring patient benefit indirectly. So you need to take into account the uncertainty that that provides. The evidence that you can put together to support use of a surrogate endpoint comes in a couple different forms. One is evidence that the biomarker is on the where it sits in the pathophysiology of the disease, and in particular, whether it's on the causal pathway. Now, C-peptide is not on the causal pathway the way HIV is on the causal pathway to HIV disease, or LDL is on the causal pathway to cardiovascular disease. Rather, it reflects damage to the beta cell, which is actually the causal factor for type 1 diabetes. Nonetheless, you want to put together a good understanding of the pathophysiology and where the biomarker fits on the causal pathway. The second is putting together epidemiologic data that demonstrates the relationship between the biomarker and clinical outcomes. And we've heard a lot about that at this meeting. I think people have done a really good job of putting this together. The third is clinical trial data, and this can be done both at an individual patient level and a trial level. We've talked about this. John pointed out some of the potential limitations, but I think this is a really important step in the right direction to get us to the point where we have confidence that a change in the biomarker will predict a change in clinical outcomes. When we have uncertainty like that, this is often the situation where we use, excuse me, a reasonably likely surrogate for accelerated approval, and then we require a confirmatory trial to show that the change in the biomarker that was used for accelerated approval really does provide a clinical benefit. Now, I want to make sure that the group understands that FDA is open to other endpoints for approval of drugs in type 1 diabetes. In the past, we've relied on hemoglobin A1C and severe hypoglycemic events. I think we have a new modality, continuous glucose monitoring based outcome measures, and promising data on C-peptide as a surrogate. FDA is open to pharmaceutical company sponsors coming in with proposals to develop endpoints based on continuous glucose monitoring and C-peptide. These are discussions that we would want to have individually with the sponsor, with the data that they provide supporting this, and the evidence that their product would indeed provide a benefit. And it is important to think about what level of change in C-peptide would correspond to a particular change in whatever the clinical outcome measure is that you're planning to use. So I just wanted to share that. I hope that's helpful.
Yeah, I think that is. John, did you have something you wanted to add?
Yeah, I just wanted to echo that point that so one thing I think is sometimes I hear is a misconception that for hypoglycemia endpoints, FDA or for new drugs, we do not limit only to severe level 3 hypoglycemia as an event. We would be open to a composite endpoint of level 2 and level 3 hypoglycemia, and in the guidance for industry, the diabetes mellitus efficacy endpoints for clinical trials, it goes into how hypoglycemia can be used in trials, and I think that evaluation of hypoglycemia is a place where CGM could be more sensitive to detect effect and perhaps more reliable than self-monitoring blood glucose, where somebody who merely treats their hypoglycemia and doesn't confirm it, it isn't captured. So definitely those are areas where we could explore further.
Yeah, Jason, I want to come back to you on some of these CGM-based endpoints, time and range, time and tight range, percent CV. I know you've been thinking a lot about this along with many others, but I'd be curious to get your thoughts on this topic. Yeah, would you consider those face valid, for example? And yeah.
So I'm glad the conversation has come back to this because I think this is very important, because we're trying to figure out how do we measure something that's clinically meaningful. And then also within endocrinology, we're looking at self-tuning systems that run within very tight parameters. And so the way I kind of see this is A1C is up here. It's a very big, broad picture. And then we can come down to time and range, and then we can come down to time and tight range. And so as our tools get better, as people are entering into clinical trials, as we're in the world of automated insulin delivery, it's harder to show changes in A1C because so many things are going into that and those things are so good. But we can still show changes in time and range, and even better changes in time and tight range. And if you go ahead and look at the world of islet replacement, that's exactly what you see. That, when you enter in a clinical trial, even before you get your islets, your A1C gets better. And then once you get your islets, your time and range gets better, but it's not as good as normal. And your time and tight range gets better, but it's not as good as normal. But when you look at time and tight range on someone who has an automated insulin delivery versus someone who gets islets, the islets are much better. And so really, I think we're kind of coning down here in what we can measure. And I would be very encouraged if we can move towards these CGM-based metrics, because they do measure these things. They do measure the ability of the system to endogenously self-meter. And that's really what we're talking about in endocrinology. That's what we're talking about here for the insulin adjustments that the pancreas is doing. And really, that's what we want to be able to measure. And I think these things give us a measurement of that.
Yeah, Elizabeth, I want to go to you. You had your hand up, and I've seen you nodding your head, and then we'll go to John. But like to hear your thoughts on this topic. Is this something from your perspective in the development world? Is this something that you think would be useful in studies? I know you guys have done a little bit of that with the PROTECT data and so on, but maybe hasn't been deployed as comprehensively to date as we know it's going to be, moving forward. So if you'd like to add your thoughts here, and then we can go to John.
Thank you. I do believe it's a very important aspect that we need to further dig into, Jason, and I totally agree with what you have been saying. I think CGM measured time in range, time in tight range is an important aspect. I think it is eventually, even in stage 2, as we heard yesterday, the presentation on that, even more important. It may help us in this area even more and sooner, rather than doing a very long outcome trial in testing new drugs. Looking in stage 2 people at the time in tight range may be a really great measure, because we see immediately the effect, and we see it at a time point where we have plenty of beta cells to preserve, which is so important, as we know. In stage 3, I think we need to really explore that carefully, because while I agree time in tight range is very important, it's still impacted by the background therapy with insulin. And we know that, right? All of us have experience in insulin studies. So we know there is an underlying impact of the background therapy that we need to consider. And I think we need to look into this a little bit more in granularity to better understand what would be the true impact on a sample size for a clinical trial seeking regulatory approval for a new drug. Maybe I stop here and I hand over to John.
John? Oh, you're on mute, John.
Sorry. Yeah, no, I think so first of all, I'd say that we do have a lot of interest in where CGM might fit in. I think the big barrier to its CGM's use in as a surrogate endpoint to support full approval in the drugs is sort of the same issue that we're talking about with C-peptide, is that you don't have outcomes data tying specific CGM metrics to clinical outcomes. And as we all know, HbA1c, why is it accepted? Well, because of these large landmark trials, and in particular, relevant to type 1D, the DCCT trial, which was 6,000 patients treated for 6.5 years with where the tight control maintained a A1C of difference compared to control of nearly 2%, and over the 6.5 years of follow-up, retinopathy, new-onset retinopathy in primary prevention was reduced by 74%, and progression of retinopathy in people with underlying retinopathy was reduced by approximately 50%. So we don't have that. However, I think we recognize that all things being equal, particularly in people who have better control, that reducing variability, improving time in range, improving time in type range is probably better. I think you still get this issue of understanding what the magnitude of the clinical benefit is, but I think I think it's face valid that in 2 patients with an A1C of 7, the one whose time in tight range is 50% is better than the one whose time in tight range is 30%. So I think those are potentially something where we could explore how those might be used. but understanding magnitude of benefit, the uncertainties, and estimating benefit risk is something that that needs to be dealt with. One point that I think is important is that, remember, with CGM, you're collecting 288 data points in one day, or 2016 data points in 1 week. So how you handle missing data becomes a challenge, and this interim missing data can be very challenging. So the categorical endpoints, where you categorize as time in range, time out of range, the assumptions that underlie how you impute those data can have big effects on the time in range. So I think, I don't think that's a problem that's unresolvable, but that is an important problem. So a different metric like GMI, or the mean glucose, the assumptions that underlie the imputation may not affect the values, and you may be able to show that or be more confident in that result than a result where you're imputing missingness, where there were a 2-hour window between two values that were very close to the threshold. So but yeah, definitely I think that's potentially an area that could be explored. And I think one big advantage of CGM versus A1C is that particularly in New-Onset T1D, is that because A1C is a lagging indicator, you get people's baseline A1C. They have immediate improvement in A1C because of tightening glycemic control, but you don't see that adjusted baseline until 2 to 3 months afterwards. And that's a challenge with that. So I think using CGM data to see what's going on before you see those changes in A1C might be part of a robust trial trying to evaluate glycemic metrics.
So Jason, I know I see your hand up there, Stephen, but I want to give Jason just a chance to kind of maybe respond to what Elizabeth and John had to say on this topic.
Yeah, no, I agree with those comments. Where the CGM is going to work the best, I think, is to be determined, and we're working on that. And then as far as how to use it, we're going to need to work out the details. There is missing data that needs to be dealt with. But all of these things are just, logistics, for lack of a better term. And so I don't think they should limit us trying to do this. This is just standardizing, and I think the field is already doing this. And so, I think it would be great if we can move CGM forward and move its acceptance forward, because as John just pointed out, it's much more reflective of what's going on now. And as Elizabeth pointed out, that lets you get to the answer faster. And then, as I pointed out, it gives you a level of fidelity that you just don't get with the currently accepted tools. And there is data that's being developed that says these metrics give more information than what A1C does. So where this data is available to look at complications, once you control for A1C, there's additional information in this. So as far as I'm concerned, it's probably better than A1C from a complication standpoint, because it isn't just an average. It includes the variability, includes all this other information. So I'm hoping that we can get here soon.
Yeah, Stephen, did you want to say anything more? And then I'm going to turn to John to kind of close us out.
Yeah, I've got two more comments on it. One is, importantly, when we think about the CGM metrics, is in the clinic, increasingly, that's what's guiding day-to-day decision making. That's what's guiding care between a patient and a physician, is not what their A1C is doing at any given moment, but what their time in range looks like day to day, what their CV looks like day to day. That's really how people are making decisions. And we need to make sure we're thinking about that in the context of a clinical trial, that we don't have too much of a spread between what we're making regulatory decisions around and what people are actually doing in real life as they think about their care. And then the other comment I want to make, John, I certainly don't think you're suggesting that the way forward with C-peptide is to do a DCCT-esque trial from here forward. I think, there's You've been very clear that that's what happened with A1C, not not what's expected with C-peptide, which is great to see you nodding so vigorously in agreement with that. And I think that's what the TOMI database can do for us. I think there, as you've said, and maybe I'm a broken record here, that we need to look at that database a little bit more closely. Maybe that also means including additional data from studies that aren't in there now, let alone kind of refining some of the analyses. But that's where we can look at this kind of bigger, higher level view of what's really happening in the face of an intervention with C-peptide and clinical outcomes. When I think about where we go forward from here, what happens after this workshop, I think, there's a couple of threads that I think about. One is just this, really diving deep into what more do we need to do with the TOMI database to really get the answer we need around C-peptide. While we do that, I think there's really important conversations happening now about this notion of face validity in these CGM metrics. That to me is probably one of the fastest ways forward here to think about improving our trial designs. I think it gives us the opportunity to do better without waiting for either a years-long new study like DCCT that we're not going to do, or just the time it takes to be able to really get to the bottom of what's in the TOMI analysis. And so I think that's really important. In the meantime, I think pushing on the PRO work, not just through PROs, but through other patient experience data, being able to bring in how these therapies are actually impacting day-to-day lives for patients, making sure that that's a part of our regulatory decisions, maybe not entirely as our true basis of approval, but as really important supporting information, supporting evidence on how we know a DMT that's preserving beta cell function, even after diagnosis when insulin is started, actually makes day-to-day life easier for patients. I think there's a lot of different things that we've talked about that are going to be pretty critical for us to collectively as a community, again, with the agency, with the researchers, with the companies, Joe, with you guys at CPATH to really pull these pieces together and figure out how we solve this.
So John, I think we're going to come full circle and we're going to come back to you to sort of close us out. And obviously you can comment on what we've just heard recently, but kind of coming back to where do we stand? Yeah. Oh, you're on mute.
Sorry, I was going to come back to one more comment that I remember that Jason had mentioned, and I think it actually does have my closing out. I think that one of the problems is that we're having a lot of discussion around how do you detect tiny differences in glycemia, and which ways are valid and which ways are appropriate. But to me, I think Jason had mentioned how what improvements in glycemic parameters we saw in islet cell transplant studies, and that made me think, I think that one of the big problems is that we need better drugs. I think we need drugs that have large treatment effects that are going to take away this whole issue of how do you measure tiny incremental differences in glycemia. I mean, in the islet cell product that was approved in 2023, this is not our area, it was in CBER. The primary endpoint was freedom from, was insulin-free days. The patients were off insulin for years in those trials. And the pharmacodynamic data that they used was mixed meal tolerance. You saw that the average patient at baseline had a mixed meal tolerance just as consistent with diabetes, where the baseline value is 180 and the peak value is 300 and something. At 1 year into the trial, the mean value was normal or close to normal. So I think one of the issues we need to see here is that we need to see better drugs that have bigger treatment effect. And I think that some of these problems are going to take care of themselves.
We also, John, we can't let perfect be the enemy of progress here, right? And we know that the treatment effect that we think about with disease modification in terms of preserving beta cell function that still exists is very different than the context of islet cell therapy. And so we can't kind of falsely equivalent these two things and say that's what we should be looking at here. Because you're exactly right. What we all want is the perfect drug that truly does cure type 1. I think that's coming. I think we see a lot of these things hinting at a true cure where you're off insulin, you don't need immunosuppression, and your life looks indistinguishable from someone without type 1 diabetes. In the meantime, I think we need to work with where we are, the therapies that we have in front of us, and find the path forward to get even incremental improvement into the hands of patients.
Yeah, but I say that I think to approve a drug, I think we have to have reasonable certainty that the drug is having an effect. And I think that if we don't know that drugs work, I think we're doing more of a disservice to people by approving something that we don't know work, where it's all risk and no benefit. And we need to have reasonable certainty that we have some benefit that allows people to make a decision about the benefit and risk. And if we can have a tangible benefit that we can communicate to patients, I think that'll allow them to be able to make those kinds of decisions.
I
Agreed.
think that's a really good way to close us out, John. I think the focus on the benefit to the patients is the right focus, and I think that's where we all live for sure. So I want to take just a minute to thank everybody on the panel. This has been a great conversation, a lot of different perspectives. Jeff, it was great to have you because you're not embedded as much in this question. You kind of have the higher level view of things, so that perspective is really valuable. But thanks everybody. This isn't the end of the conversation. It's, as I framed it at the start, it'll be the catalyst for the follow-up that we need, and hopefully we can shed some more light on this question of C-peptide proportionality and help with some of the additional analyses, John, I think that you and others there at FDA would like to see. So thank you again for everybody, and thanks for the great engagement and discussion.